So I'm delighted to be here with my very old friends, two of whom are right here in the front row, and they're more scattered around. And the timing is good because I'm just one of the many in the midst of thinking about working on the President's Precision Medicine Initiative. So I'd actually like to go fairly rapidly through my talk because I have the vague sense that there's plenty to talk about together here in our short time. The Dean and I did have a wonderful conversation earlier, in part about the role of genomics in public health, so lots to debate. And what I'm going to use as my linchpin is an area that I've been working on for a good 10 years. But what I want to do with this is something new, which is move it all the way out to public health benefit. I've been involved in this debate, which is really no longer a debate. It's now a consensus about the importance of giving people back their own information, which, by the way, is one of the core commitments of the Precision Medicine Initiative. So as I think our group actually got the first NIH grant dedicated to this problem that started in 2005. So having sort of been there through the history of the evolution of this problem, it's amazing to see it enshrined in the Precision Medicine Initiative documents to date, that all participants are entitled to return of all of their data rapidly, in addition to the scope of interpreted results, personal results that they consent to. People can elect not to have any of them or elect something in between. The precise architecture of this has to be rapidly determined as this thing techs up. They're also entitled to the results, the aggregate interpreted results of the cohort, of the full cohort, and exactly cashing that out is going to take some work too. But there is intrinsically here a two-track vision, a vision of the individual and a vision of the cohort, both operating at once. And part of what I'm going to be arguing is that they are very closely linked, that the translational process that we're seeing play out in the space I want to talk about, which is largely about genomics, deeply links the two. We have long been in a sort of nature-nurture, either-or debate. I mean, as a scientific matter, we know that that's wildly outdated. We're still debating it on some levels in terms of where the money should go, where the effort should go. But what I want to argue is that we're at a pivot point. I hope we're at a pivot point where we are deeply linking the two, and the question is whether we're going to do it right and whether we're going to do it well, so that we can be progressing both at the individual level and at the population health level. So I'm going to use this information transfer issue that I've been involved in for a decade as a way of opening up a broader set of issues here. Here are my disclosures. Return of results has become shorthand. It's shorthand for, are we going to give people back their information? It's not a great shorthand, although people like the shorthand ROAR. But it's not a great shorthand for a few reasons. First of all, it covers a lot more than results. And also, return sounds like, darn it, whether you want it or not, we're going to give it to you. And that's not at all what's going on. There's a little skirmish, and George and I have been involved in this skirmish about whether to force results on people, and I'll get to that briefly. But really, this is about offering this information to people. It also could be misleading a little bit that I'm talking about it so much in the genomics space. I'm talking about that because I was asked to talk about public health genomics, that's one word for it, really in the precision medicine, et cetera, this issue. But this problem of return of results cuts across every form of research. And by the way, it cuts across every form of clinical encounter as well. There's always this question of what, if anything, to tell the patient or the participant or the source individual for biospecimens, what to tell them about what you've got and what you've gleaned from their specimen, from their data, from their examination, et cetera. I bet everybody in this room has had the clinical experience. I've had this clinical experience. If you go into the physician in a clinical visit, and you say, I've got this problem, and the physician says, well, what is that? You know, maybe something dermatologic or something else that they see. I was just recounting a story to George and Wendy earlier of being worked up for what turned out just to be an atypical presentation of a migraine, and they come back and they say, which turns out to be wrong, which is another issue here, you have a meningioma growing in the middle of your skull, which I didn't, as a matter of fact, but it was an incidental finding. So there are all these issues in clinical encounters and research encounters, and by the way, in public health surveillance, but what do we tell people? How much confidence do we have? My little vignette raises that in the accuracy of what we're telling them. What are we suggesting they do with this information? And actually, what do they do with this information? So there is a cascade of questions around this information issue. Dr. Collins, there was a front page article on this whole issue a few years ago in the New York Times, called this cluster of issues one of the thorniest challenges in clinical research. And I think that that's proven accurate. As a funded investigator, I participate now in the Clinical Sequencing Exploratory Research, which has another cute acronym, CSER, Consortium, which is a set of funded projects involving, altogether, we're about 277 investigators looking at genomics and looking at return of results. So this has become a very big deal. And it's not just this consortium, there are a bunch of other consortia looking at it too. There's been an outpouring of research and scholarship, and even, here's my other little nugget, of evidence that we're moving into prime time, not just that the PMI says this is a core commitment, but actually in the embattled, is that a good word? Embattled notice of proposed rulemaking, pending, long pending, to amend the common rule. We don't know where that's going, but it even crops up there. For the first time, the common rule, which, as most of you know, is the DHHS and about 18 other federal agency rule set for governing research involving human participants, and the proposal, among many changes in this big proposed change, is to talk directly in the proposal about return of results, where they say that if you thought you were exempt or if you thought you were excluded, no, if you're doing return of results. It ratchets it up. You've got to go to your IRB. And at core, what's going on here is a fundamental tension that emerged, an empirical tension that if you ask people, do you want to know what we find? What do you think they say? First of all, they look at you like you're nuts, like, oh, you were planning on not telling me? And then they overwhelmingly say yes. The data are more nuanced than that, because if you ask people not just out on the street, but people actually in projects or people affected by a certain disease, and you start to say, well, would you want to know about highly clinically actionable, highly pathogenic stuff? They say, yes, way up in the 90s. But then as you go down, what about carrier status? You know, it's not going to affect your health, but it might affect your offspring. It might affect your reproductive decision. You're still above 50%, but you're not in the 90s anymore. If you go all the way down to variants of uncertain significance or unknown significance, VUSs, it gets softer still. Though, you know, overwhelmingly people say yes, really across all designs when we've asked this question, but the historical custom has been not to return. So I'm going to talk about that from research to clinical to public health. Okay, obviously this set of issues is all the more important because now we're talking about launching the Precision Medicine Initiative to establish a prospective population cohort of at least 1 million participants. And this is a very, very ambitious proposal. It's easy to mistake this proposal, especially with that much publicized picture, which is a very nice picture of the president, but he's sitting next to a big molecule model of DNA. It's easy to mistake this as genomics. It is much broader than genomics. It's about collecting all sorts of data, including self-reported on telephones, smartphones, in all sorts of ways, empowering citizen scientists, that's the vision here, all the way to exposure data. So it is physiologic, all sorts of information. and environmental, not just that person, but what are they actually being exposed to, ascertain in a way independent of that person's report. So it's a very, very ambitious project to assemble a truly big data set, but also to get and keep, which ain't easy, as this crowd knows, very high participant engagement over a long period of time to assemble a longitudinal data set. And how to do that at this scale in this country with the other pillar of this project, which is diversity. Genomics suffers from the fact that we do not have adequate data sets, flat out. They are too white, they are too unrepresentative of our population across multiple vectors, money, education, et cetera. And what that yields is inadequate data with which to do pretty much everything. Predict risk, interpret genomes, and the like. We don't even have adequate data sets in terms of healthy people walking around the street, because overwhelmingly, these data sets are affected people. So there was a recent article by Van Driest et al. In JAMA that revealed a lot of problems that grew out of the eMERGE Consortium, which is another one of these NIH-funded consortia. And one thing it revealed is they took the same genomic data set and they gave it to 3 superb labs, and they didn't agree on the variants they found, the genomic variants, and called. There's a problem. And also, this problem of unrepresentative data sets emerged because it was clear that what they did call was more inaccurate for the populations poorly represented in the existing data sets. And it was also clear, this was in cardiovascular risk genes, it was also clear that they did a very poor job of telling you who was sick and not. Of course, now some of these are predictive genes, but they took these genes and they looked at unaffected people and they said, well, given what we're seeing genomically, we should be seeing something on your echo, or, and they looked and they looked and they looked. And for a lot of these people, they couldn't find anything. And this is a very common problem across genomics when you're operating from inadequate data sets that really are affected people and don't tell you all the full spectrum that you need to know about penetrance. And maybe people can walk around with this gene that you thought in affected populations would mean you dropped dead, or you were severely affected, but it turns out that it's much more variable in expression and much more variable in penetrance. And so the people, and the dean and I were talking about this earlier, the people who I find who really do genomics day in, day out, know all this. They do not overestimate the data sets. They do not overestimate the science. They recognize that in many respects this is early days and are all the more intent on making progress with this. The Precision Medicine Initiative in some ways grows out of a National Academy of Sciences report in 2011, I believe, that really was even more ambitious, thinking not just about all these data types, but thinking about a transformed ontology of disease and illness and risk. Because that's really already happened, as many of you probably know better than I do, chapter and verse, in cancer, where we now really understand cancers as dysregulated pathway diseases, where it now makes sense. The BRCA gene is not just about breast and ovarian. Turns out it has something to do with pancreatic. It has to do with all, P53. I mean, you name the gene, depending on where it is in the pathway. We now understand that the, ultimate organ that ends up affected is just the ultimate organ that ended up affected. That upstream, there was a lot genomically going on that we really need to understand. Okay, so as I said, I'm going to talk about across, in a translational way, across research, clinical, public health. And this is just a quick summary of our work. So let me start with the research sphere. So just to make sure we're all on the same page, these definitions grow largely out of work that I've led. So the structure of the work that I've led is empirical and normative, but the punchlines that I'm going to focus on here really grow out of normative national blue ribbon groups that through a structured process generate consensus guidance. which usually takes 2 or 3 years. We just finished a project on the unbelievably complicated problem of offering probands, offering an individual's results to family members, including after the individual's death. And as you proliferate the number of characters in the play, and you get more deeply involved in the guts of HIPAA and the state analogs and everything else, my goodness. So that one took 3 years to achieve consensus, even with very, very experienced people. But when we're talking about an incidental finding, just so we are all on the same page, we're talking about a finding about an individual research participant, a finding with potential health or maybe reproductive importance, or we might even say now personal importance, because now we've really kind of trifurcated the spectrum. For example, you might say APOE4 variant, it's pleiotropic, but to the extent it reveals risk of developing Alzheimer's, we can fight about it, but there's not that much of proven efficacy that we can do yet. But people may still very much want it for their own life planning, for their estate planning. People may also very much not want it. There's James Watson, who famously underwent whole genome sequencing when it cost a lot more than it costs now, said that the one variant he didn't want, reflected in his public posting, though it's been imputed at this point, was his APOE4 status. So some people don't want to know that that may be looming or at higher probability in our future. Individual research results, in contrast to incidental findings, these are the variables that you're after as a researcher. These are the variables that you wanted to study, but in a particular individual, it may be that their results lead the investigators to think, my goodness, this has real actionable health implications. Shouldn't we be offering this back to somebody, to the person, to their clinician, to somebody? And so that's that problem. And then aggregate research results. That's the stuff you intended to publish on your cohort or your sub-cohort. But there's bleed among these categories. For example, I frequently collaborate on one project with colleagues who run an NCI-funded pancreatic cancer biobank at Mayo. And of course, pancreatic cancer remains a very bad cancer from the standpoint that median survival from diagnosis is still only about 6 months. And so they published in their newsletter, and for biobanks and biorepositories, family registries, which is the species that Mayo is, it's good practice to be communicating with your source individuals. And so they published a newsletter saying, we've discovered that this variant that was thought to be associated with melanoma risk seems to have some role in pancreatic cancer risk. And sure enough, one of their participants showed that to his physician, and the physician said, well, go back to those people and ask them, are you one of them? So that was trying to move from aggregate up to individual. So there's play among these. Now, in the Clinical Sequencing Exploratory Research Consortium that we participate in, this is a hard-to-read map about all the participants. I just want you to look at the yellow highlighted column, just to get a sense of how frequently these things show up. So this is about percent of participants with at least one finding relevant to their phenotype. This is in research. This is a research result, not an incidental finding. It's relevant to the phenotype that's being studied, which is why I'm calling it a research result. And you see that it depends. So just look down that one column, pathogenic or likely pathogenic. And I could go on and on about why am I saying pathogenic and likely pathogenic, but I won't. Some of you already know. But I just want to get to the punchline that there's a big range here, from 1% to 39%, depending on which project you're talking about. Is it all cancers? Is it cardiovascular, all the way to ophthalmology? So that gives you just a feeling of some of the numbers here. And they contrast some with the incidental findings. Now, again, it not only depends here when you're talking about how often you encounter these things, what population you're talking about, it very much depends, as I'll get to, whose list you're using of incidental findings. You would think there's an agreed list, and if you go to the NHGRI website, you will see a non-endorsed, it's carefully framed, a non-endorsed list of the incidental, or sometimes they're called secondary findings, that CSER projects and Emerge, and I think NSIGHT, another consortium, are offering back to people non-endorsed. And I'll talk about these various lists. But you see that these numbers before you get to carrier status are still pretty small, but it very much depends. If you're going to do a whole genome sequence and look for potentially everything that's pathogenic and actionable, particularly if you're going to include carrier status, everybody in your cohort will have one of these. Okay, so there are a whole lot of questions that spill out of this. You're already getting a sense of some of them. What's a subset we should worry about? Maybe we shouldn't make it a subset. Maybe we should, and there are technologies for doing this, offer the whole set to people. Here, click on this. If you want everything, bless you. Here's your whole set. Good luck finding somebody who can interpret the whole thing. Nobody's going to say that last bit, but I'm going to say that last bit. It's hard. How are you going to plan for this? How should the whole thing work? And this is part of the challenge of the Precision Medicine Initiative, too. Now, I'm not going to dwell on this because I've long made this point, including in print, but this is an intrinsically translational problem, and I'm going to extend this. Here I'm simply arguing that it's translational in the sense that you've derived these findings in the research context, but the whole reason you're getting worked up about them is because they have potential implications over in the clinical context, which is not the context you're collecting them in. In fact, they may not be clinical grade. One of the skirmishes, the side skirmishes, although it's important in this, is do you have to have a CLIA-certified lab to find these things in the research context to offer them back to people? Or can you offer them back to people saying, hey, red alert, it's not from a CLIA-certified lab, it's not clinical grade information, but we're worried enough about it to give it back to you. You go, work with your clinician. It should be redone in a CLIA-certified lab and maybe validated, orthogonally. It's all that stuff. But we're worried enough about it to tell you we may be wrong, we may be right. Some people worry whether that's okay, ethical, and by the way, legal. That's the CLIA fight. So there's a lot going on here. I hope what you're hearing in that is the sort of epistemology of this problem, that the kind of information you may be generating in research may be quite different from the kind that you're generating in the clinical space, where we have stricter rules because it's the clinical space, and yet we want to hand this off. This is a slide I've used for a while too, just to demonstrate, I'm not going to go through it all. Not only do we have this sort of epistemology, what's the caliber of the information problem, we have very different norms operating in these two spaces. Yes, there are domains like oncology where we commonly mix both, but that's actually a problem, not a solution. It's a question, not an answer. So it is tricky to move this information from the research space under the microscope to the caduceus space, to the clinical space. One really stark example I had of this very early in my trying to wrap my head around this problem was not from genomics. It was from imaging research, where, when this problem first came to me, a very dear colleague of mine, then at Minnesota, now across the river at Harvard, said to me that he was doing imaging research with normal adolescents, really cognitive neuroscience, where he'd put them into magnets and he'd do some structural stuff just to map what's where in this kid's brain, and then he'd give them puzzles and see what lit up. I mean, that's my, the lay version of what they were doing. And a few months after doing this with one of the kids in the study, the kid had catastrophic seizures. They worked the kid up clinically, and there was a big old brain tumor, and the parents said, what the heck was going on over at the U, as everyone calls the University of Minnesota? Didn't they just look at your brain? Well, the answer was yes, they just had looked at his brain. But the reality was it wasn't clinical grade scanning, and it wasn't telling the magnet, the MRI machine, to do the things you would do, to run the sequences you would run, if the purpose was diagnostic based on some clinical indication or suspicion. Nonetheless, when they went back, there it was. But it gives you a sense of this grade issue, what I'm calling this grade issue in the epistemology of this thing, why it's so challenging and why it's deeply translational. It comes up too in biobank research. I think in some ways our most contested consensus paper was, our first one was about incidental findings, so that was with one whole group. And then we moved into the biobank world because the reality of genomic and genetic research, and much research now, including much epidemiological research, is very large data sets, archived data, archived specimens, reanalyzed by groups, maybe long into the future. And so the question is, what do we do? I mean, the biobank community, much of it was saying, don't look at us. We are not set up to be going back and giving anything to anybody. We are set up, this is one cartoon of one way, there are many ways in which you could organize a kind of biobank research system where you have multiple sites collecting the data and or specimens. It's all being fed into the mothership, the green hexagon. That's the biobank, and that's the people saying no. And then they're saying our job, and by the way, our budget, is to organize this stuff expertly, to do QC, to make sure we know what we've got, and then to feed it out to the red squares, the secondary researchers. That is what we do for a living, and many of us get fully de-identified stuff. So who do you think we're going back to? Anyway, we're not holding the codes, and because we're not holding the codes, we are not doing human subjects research under the common rule, by the way, et cetera, et cetera. So we've relieved ourselves of much of this burden. And our argument, and it met with a lot of resistance, was sorry. You are a central feature of a whole biobank research system that's organized by contracts, among other things, material transfer agreements, material data agreements, et cetera. And the whole system should agree on what is the game plan. If you are collecting people's data and specimens, people have to know. What's the plan? What are you going to do with it? And this argument that it's de-identified, and not all biobanks do get the de-identified stuff. Sometimes it is identified. So what? It's re-identifiable either at this site or sometimes there's a trusted intermediary. So we made this argument, and I think have largely succeeded with this argument, though not with all biobanks. So it affects this kind of setup too. Just to give you a feel, our recommendations and many other recommendations since have kind of divided the problem into three sets. What should be offered? So should return is that return of results shorthand problem. What should be offered to people, because consent, every time we have gone after this problem, it hinges on the person having the right not to know, and the person retaining full autonomy, full consent rights, full control over this. But should offer, may offer, and then there were some don't offer, like, really? You have misattributed paternity? What is the point of offering this back at this point? You can come up with scenarios where it might be relevant to something, but there are a lot of scenarios where it's not relevant to something, and so you're just going to risk blowing up a family, and it's not clear why. So there's been change, I think, over time. Over time, should return, in many of the recommendations, has become focused on highly pathogenic. We have a good deal of confidence in this finding, even though it's a research finding. It's clinically actionable. The people in the white coats feel that they can really use this to change diagnosis, treatment options, prognosis, outcomes, and you've got consent, whereas may is kind of everything else, including carrier status, reproductive importance, in other words. I just was at a CSER meeting and participated in a panel on carrier status. You know, the truth is, all of the CSER sites, except for 2 at this point, are returning carrier status. The scope of what they're returning varies in terms of what carrier status are they actually returning. But I think the pendulum is moving, and we can talk about whether that's good or bad, on carrier status and reproductive importance. I happen to think that's progress, but we can talk about it. Okay, and then there's this new killer issue, and we just came out in the Journal of Law, Medicine, and Ethics last fall with a consensus paper, and then a lot of other papers around that, some empirical, some normative, on this very difficult tension between the reality that we largely approach ethical and legal rights, including privacy in this country, as an individual matter, as a personal matter. But the reality is that if you're talking genetics, there's this huge familial dimension. And so in that pancreatic cancer biobank, you get families coming back and saying, well, you know, Dad passed away. What about our risk? Huh? And what else have you got behind the doors? So it's a very important domain, particularly because a lot of genomics really has had first rollout in the cancer space, life-limiting illness, and then you've got long archived data and specimens, and the family is showing up. Or in some cases, the researchers are saying, we want to reach out to the family. So I'm not going to dwell on the guts of our recommendations. I'm happy to talk about them if you want to in the questions. So that was all talking about research. Let's go clinical. And this is an area that George and I have written about. This is probably the most famous paper trying to look at the clinical genomics space. If you think about genomics, particularly in the context of whole exome and whole genome sequencing, even though you're not doing research, there is this problem again of drinking from the fire hose. So let's say you are sequencing a cancer patient because they have failed, they're no longer responsive to conventional therapy, and you are looking for something else to do to save their lives. You're looking for a new molecular target. There are other scenarios. You've got a kid with a very puzzling neurodevelopmental anomaly. You've done the usual diagnostic odyssey. What the heck is wrong with this kid? And so then you may resort to sequencing. You may sequence a trio, them and the parents, to figure out what is going on with this child, and is there anything we can do about it? So there are a lot of different scenarios where if you're doing a whole exome or a whole genome, there's all this other stuff, and the question is replicated again. What are you going to offer back to people in a way that makes sense and that's responsible and that doesn't drive them crazy and that respects their preferences? So what's initially appealing and then disturbing about this important paper from Jonathan Berg and Jim Evans at Chapel Hill is how similar it looks to what I just put up for research. Should, may, don't. And there's all hinges on actionability. Why is this a little bit disturbing? Because this is the clinical space. And you would expect the standards, I would argue, to be even more respectful of the question, what do people want? After all, these clinicians have as their first obligation caring for this whole person, not just their genome, not just their arm, but this whole person who has values and wishes and preferences about informational return. And yet, this hinges entirely, almost, on what can the people in the white coats do with this information. And I think that's a vulnerability of this scheme. In their defense, this was 2011, and it was progress to even get this out there. Then there is an argument that this is the core of what George and I with Sherman Elias published on. The American College of Medical Genetics and Genomics is the professional society of medical geneticists. So they dug into this problem starting with 2012, where initially they said a good thing, which is, okay, red alert, big problem. So they're really speaking to labs and medical geneticists and also some genetic counselors, though the genetic counselors have their own professional society. So they say, once we do sequencing, which we're now embarking on, incidental findings definitely are on the horizon. So you got to get policy, you got to tell people in advance, you've got to let them say no. Like maybe the cancer patient that we hypothesized says, you know what, I'm fighting for my life. So anything you find about my cancer, cool. I want you to have it. I want to know about it. I want us to use it. Maybe I want to fly to MD Anderson. I don't know what I want to do. I want the cancer stuff. But if you're going to like troll the countryside of my genome, looking for all this other stuff, hold it. I'm not there. I'm not with you. Or, maybe the person would have a different preference. But this was saying whatever their preference, whatever their circumstance, elicit and respect it. That caved, however, because the very next year, ACMG came out with the proposal that, you know what, whenever we're doing sequencing for any indication, the cancer, the kid, anything, you name it, whatever we're doing, tack on some extra analysis. Because we have come up with a starter list, just a starter list, of 56 extra genes that are so big, bad, and ugly. And I don't mean to make light. I mean, this is stuff like BRCA, Lynch syndrome. They are big, bad, and ugly. I mean, they can be addressing real risk in this individual. And highly actionable, those were their real criteria, pathogenic, highly actionable. Whenever you're doing sequencing, treat this as opportunistic screening. So I'm purposely now transitioning into some of the language of public health, and that was the language they used. Opportunistic screening. You got them right there. You got the sequence right there. It was really paternalism. We can save lives. That was the real argument. And I really don't want to make light of it, because that is, in seriousness, what they were trying to accomplish. And remember, this is the clinical space, not the research space. But the problem was that the way they did this was saying, don't give people choice. They're going to opt out of some of this. And then what? They could die. It's like, yeah, they could, but people do have the choice to not be burdened with this. In fact, people refuse life-sustaining treatment. People make a lot of choices where the consequences could be fatal. And by the way, this is risk information. It's not like they're going to drop dead right in front of you. So nonetheless, they said, we do not favor offering the patient a preference. This may be seen to violate existing ethical norms. Yes, in fact, yes. Like autonomy and the right not to know, which has been a kind of axiom of genetics for a very long time, but that's what we're doing because we're doing opportunistic screening. We can achieve individual, and the implication was even some population benefit. Firestorm, 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 this paper that we did together, George and Sherman and I, and then was responded to, was just the tip of the iceberg in terms of the firestorm. And to their credit, they then switched gears. Now, this was April 1, 2014. I briefly entertained the possibility that this was some kind of April Fool's move, but actually it was a real move. They didn't fix the whole problem. But I think actually the ACMG deserves a lot of credit. It's hard for a professional society to put a stake in the ground and then go, you know, we weren't quite right. That is hard to do. It's unusual. So what they said is, really, there should be an opt-out. Now, what they didn't say is there should be an opt-in. That would be even more respectful of people's wishes. Nor did they say, well, what about our cancer patient? I mean, can people pick which subset they want and they don't want? And also what they didn't fix was kids, where they were saying, oh, yes, they even do this in kids, even though it's long been a tenet that really we don't do genetic analyses in kids about adult onset disorders. We let them decide that themselves when they come of age. That's sort of the dominant MO that we've taken with kids, and that's not fixed. Okay, public health. So there's already a lot of discussion about the way this problem erupts in public health genetics and public health genomics. So Michelle Lewis and Aaron Goldenberg published a paper, for example, in our Journal of Law, Medicine, and Ethics set from last fall about newborn screening. There's a lot of discussion now about how broad to go with newborn screening, whether actually to go exomic or genomic with newborn screening. And also, when you do newborn screening and you archive the dried blood spots, which you may do for de-identified further research, first of all, for quality control for your program, but also for further research, what if you discover some urgent finding in that context? Who are you going to tell? And there's been actually a lot of litigation in this space. The good people of Minnesota and Texas brought suit saying, don't you dare archive and do research on our baby's blood spots without our express permission. They prevailed in Minnesota. The Minnesota Department of Health had to destroy 1 million Guthrie cards with blood spots, a giant population study resource. It was anguishing to our public health colleagues and people all over the country. The Texas case, I think, was settled. And there have also been cases brought where people said you were doing pilot testing in the context of newborn screening and you didn't give us our results. And some of that has been successful, whether settled or through court decision. So there's a little litigation worry going on here. And a little bit of state statutes like South Carolina starting to say, you can give back the results. So this is interesting to me because this is the public health side of the problem. In the context of expressly public health functions, what should we do about the information? Who should we offer it back to? I was asked to consult with a bunch of other people on the NHANES project, so that should be familiar to a lot of you who do public health, big federal health, nutrition, et cetera. That's the Apple. Cardiovascular, et cetera, project, because they were collecting genetic information and archiving it too, and they were thinking, what do we do with returns of results and incidental findings? But part of, I think, the answer here is when you are talking about public entities, this responsibility to deal responsibly with the information and to be actually responsive to people's desires and to ascertain those desires for information back is heightened. So are the budget constraints. The budget constraints are heightened. But I think the era of ostrich in the sand, all we're doing is research, or all we're doing is public health, so we really don't have to worry about those people beyond the narrow scope of what we set out to ascertain is probably largely over. And it's partly over because people are suing to make sure it's over. There's also a very interesting debate about return of environmental exposure results. A new colleague of mine, I hope she's not in the audience because I'm going to refer to her work, Julia Brody, who's at the Silent Spring Institute, who's done very, very powerful work about exposure to endocrine disruptors and other environmental exposures, some on Cape Cod, some elsewhere all around the country. Wrote a wonderful article that you can't quite see the site there, but that's the beginning of the title of it, collecting what the environmental folks have written about return of environmental exposure results. And you get some of the same problems. Well, we're not sure what it means. We're still studying it. Should we offer it back? But still a huge move in the direction of yes. So what has emerged, and part of what the dean and I were talking about, and I know Muin Khoury, I think it was here, or you all had a debate quite recently, has been one of the people talking about a vision of public health genomics, in which a lot of things are already going on, determining when genetic and genomic tests are ready for screening application. There's a very important group called EGAP doing that. Obviously, doing the age-old task of distinguishing what's causing what role are genes playing, if any, what role are environmental exposures, which exposures, what role are obesity, inactivity, et cetera, playing, in how can we prevent this, trying to really disaggregate this, targeting prevention efforts by susceptibility, genomics of pathogen and host, modernizing surveillance by importing some of the big data stuff, or, it's really bidirectional. So let me make my last point, which is the translational nature of this whole problem. This is a very familiar picture. Actually, Muin's group did this, but it's one of, if you Google translational science or translational research and just look at the images, it's like there are a ton of these. Trying to visualize T0 to T4, this progress, so that by the time you're getting over here, you're talking about population health measures, but it's trying to map and think systematically, particularly in this era of CTSAs, et cetera, about this whole progress. What's been greatly missing from this is the ethics, much less the law, of translational science, and a sense of what are the best practices for moving around and integrating in this sphere. So I recently published a paper with Barbara Koenig and Wiley Burke trying to fill that frame and talk about the very interesting problems that erupt from the reality that you don't have bright lines and rivers and walls separating anymore research and clinical. And by the way, research, clinical, and public health. We know, yes, when we're all the way in public health surveillance and public health screening, but there's an awful lot of permeability at the boundary and translational progress. So problems arise. Well, which set of norms should apply in this context? For example, the CSER projects, which really straddle research and clinical care, you get into these amazing conversations. People go, well, so where should the results go? Should they go in the research record? Should they go in the medical record? I know of one project that asks participants, where would you like your results to go? I know of another that created a third kind of space in between. It's just one piece of evidence of how tough it can be to straddle the line. You can't see this full thing, but I just want you to get the idea that one thing that Barbara and Wiley and I put forth is what are all the ethical frames, because there are multiple that have to apply as you go around, and tried to cash that out. Everything from the ethics of human non-subjects research, as it's been called, to human subjects research, to biobanks and data archives, to public health, to organizational ethics, to the ethics of learning healthcare systems, something that the Hopkins group has written about, the ethics of outcome assessment. You have to integrate all of this really and think in this kind of big term about it. And we actually proposed a decision rule for what do you do in the conflict spaces where, like CSER, where you really don't know which set of norms apply. But part of what I want to argue here, and part of what at least we argued in the paper, is that one of the interesting things about this information sharing, this information return problem, is that in some ways it's motive, it's motoric, it's dynamic. It moves you around the circle. If you're in the research space, you're asking, well, wow, maybe we should move some of this over into the clinical space. Or if you're in the clinical space and you say, well, maybe some of this is ready for opportunistic screening or more deliberate screening, and you're moving into the public health space. And all of these are examples of information return and the pressures of we have something we think is valuable informationally here, driving you to pave that translational pathway. So I hope I've begun to persuade you that this problem, which initially looks narrow and looks arcane about what should we give back to people, isn't. It's big. It cuts across all of these domains, and it's actually, I think, doing something interesting in moving us forward around the translational cycle. What I would still argue in my last bullet is that the core challenge across domains is how do you advance the science, the biomedicine, and by the way, the public health, while maintaining respect for the needs and the choices of research participants, of specimen sources, of patients, and of populations, engaged publics. Thank you. So I think I talked fast enough for at least some time for questions. Yes?
Thank you. Yeah. Hi, my name is Richard Sates. Nice to meet you, and thank you very much. I wonder if this, I'm thinking about prevention and clinical screening or clinical prevention as the model, and we have very high standards for that. And I'm wondering if you think that the standards for return of information should be, or of results, sorry, should be less. And the reason I ask that, there's a tendency, I think, for people to believe that more information is beneficial and good for them, right? And yet, knowing the results of ovarian cancer screening, for example, is likely more harmful than good. Knowing the results of prostate cancer screening is likely more harmful than good in terms of outcome. And most of what you discussed in terms of value of information, I believe, was about rights and about knowing and maybe knowing something about prognosis. The word actionable came up, but usually the actionable part in the information you're talking about, I believe, remains unproven. And when we go to do large trials like prostate screening, ovarian cancer screening, even, frankly, breast cancer screening, which has tiny, tiny benefit, right, if at all, we at least know the risks and benefits, and I think here we may not. So I'm wondering if you're suggesting that there are different standards from what we usually do to large populations and should be because of the value of this information, or if we should be dealing with sort of similar standards.
So I think it's a great question, and EGAP, at the CDC is addressing some of this. They've got these tiers. How much evidence do we have of net benefit from offering this information? But I think you're totally right that it's early days in terms of applying the kinds of screens you're talking about, which are the same kinds of screens that they're talking about. So I think largely we've been talking about the return of research results on an individual level. And we're only beginning to collect the kind of data you're talking about. And we're actually really struggling, too, with the question of what does the post-return healthcare utilization profile look like? Because that's another dimension of this. First of all, you can give something back that's just wrong. I mean, I take your point even if it's right, but it can also be wrong. And there's some famous cases of people incurring very burdensome, and by the way, expensive, but also just burdensome, follow-up procedures with their own risks and their own sequelae, and then it turns out there was nothing there. It was a false positive. So I think economists are showing up at these meetings more and more and trying to think about and devise ways to really rigorously look at the utilization profile too. So there's a big cost issue. And that comes in because the payers are looking at this and going, wait a minute. So let me get this straight. You got this idea that you're going to do good by giving out all this information, and then what is it you think we're going to pay for? The pursuit of all this stuff? Think again. We have actually our own filters, our own screens for when we're willing to pay for any of this. So that's in the mix too. Yes?
Thank you, that was terrific. So let me start with a specific question. So APOE4, is that in the required reporting or is it in the May report?
It's usually in May.
So it's usually May. So the 56 genes, right, it strikes me that the entire thing falls apart or stands tall based on who's defining utility. Because, I mean, you mentioned BRCA, which is fine. I mean, there's plenty of argument about whether BRCA, about the positive predictive value of BRCA, so what utility means. And I presume this is being determined by the bodies that you mentioned. So can you educate us a little bit, like where is the argument about who is determining utility and the consensus around what decides that something has utility? I would imagine that there are many pharmaceutical companies who would argue that APOE4 should actually go into the level of has to, based on some sort of dodgy results, but it's certainly just where the world is headed. So where do we draw that line, and who is ultimately the arbiter of what utility is, and who should it be?
So when ACMG came out with the list of 56, what they said is we have a committee, and we met for a year, and we achieved consensus. And then a lot of people said, based on what? I mean, that's nice. And Wiley Burkle had a paper that I was on that basically said, based on what? And also said, opportunistic screening, and this relates to the prior question, there are rules for that. You need an evidence base. You don't have the evidence base yet, so it falls on both scores. And it's even a little worse because the ACMG vision was, here's the starter set, but if labs, individual labs, want to add to the list, sure. And so that's not stating a methodology. And some of the CSER projects, they're way north of 56, 114, there are different lists being offered. So I don't think there really is a rigorous methodology, and moreover, there's this sort of, in some ways, small p political problem you raise of who's in charge of this? Is it just the professional society of the medical geneticists? Because part of what you heard in my presentation was implicitly a criticism of defining actionability only from the standpoint of the medical geneticist, or even only from the standpoint of the clinician. Because the individual may say, well, the heck with that. I actually want it. It would be actionable to me. I mean, I might do something different. By the way, my action might be that you're talking about me. And so if you tell me, well, we don't fully understand this finding yet, that's nice. I will follow the literature. I have the capacity to do that, or there are apps being developed that will help me do that, et cetera. It's not you, and you're not promising to let me know when something new comes out or when there's some new publication that clarifies that, yes, you know, this variant is associated with this probability with this, and we can do something about it. So I'm going to follow it. I'm the one with the biggest stake in that. So there really are big problems. The other problem, though, is this problem of choice, of saying, we know best, and we're going to do this whether you want it or not.
So two follow-up questions. So I noticed that and I don't know this field at all, so it's very instructive, thank you. It's not just health, it's also reproductive significance. It strikes me that the question of utility in terms of reproductive significance is even slipperier than it is around health. Because of course, what is important to me, what has utility for my reproductive concerns might be quite different than what's important for you, for your reproductive concerns. So I was wondering if you can comment on that. And let me ask you my last question, and I'll be quiet. So where is the field at in terms of then retrospectively going back and giving participants? Because today it's 56. The ACMG Committee of Smoke and Mirrors meets tomorrow, and it becomes 59. And are you telling me that you're not going to give me those 3 extra alleles because I happened to be screened yesterday but not tomorrow?
So first of all, I don't want to say smoke and mirrors, because these were actually very, very thoughtful people who were trying to do good and who have been, you know, have stuck in there, even though it's been hard to hear this level of criticism to what they produced. So very thoughtful people. The retrospectivity issue is hugely difficult. And so I was just talking with one of your colleagues, for example, about Framingham. I mean, these long-established cohorts, now what? It's not just that we may know more because we may be undertaking more sophisticated genomic or other analyses, but really the pendulum has swung in the direction of this returning information. A lot of the studies I see trying to deal with this reconsent everybody. So they don't just call the people who have a bad finding and say, hi, we know something. Do you want to know it? And don't get alarmed. You know, it's like impossible to do. I mean, I have a Pandora's box right here, and don't worry about the box. It's just, so it's easier in some ways and probably more ethical to just try to reconsent the whole population, et cetera. But it really is an issue. This is much easier to design prospectively than retrospectively, and there's a big recontact issue. So even if you're designing prospectively, are you going to go back to these people? So 3 years from now we know more? You know, that's like my newsletter problem, where, you know, in good faith we sent out the newsletter and now they want to know individually. Reproductive, you know, it's a hot potato. I mean, we are in the land of wildly divergent, not wildly, but deeply divergent views and preferences. We are in the land of potential termination of a pregnancy, abortion. We are in the land of reproductive planning, and maybe I shouldn't use my own gametes. This is really hot potato stuff. My own feeling is this is a large part of why reproductive, though in the early recommendations it was up there and should return, went down to may return, like let's put it in may return. I also think, in honesty, that it was demoted because people started to think, carrier status, you're joking. That's everybody. We can't possibly do this on that scale. Now we're a decade out and we sort of do see how to do it, at least in particular. But you're right, and particularly when I talk about carrier status reproductive, then I do not say return of results. Offer, highly optional offer of results. But it's interesting, I think I mentioned one of the Caesar projects, this is the entirety of what it's doing. Part of my argument about why it's important to look at reproductive results is I think it has actually a differential impact on women. That, you know, we have rightly said we have red alert, giant, urgent problems with the lack of diversity in our data sets. We also forgot about gender or sex. That, you know, actually the impact for a woman who has to undergo more invasive screening and maybe face a much tougher situation is it's not symmetrical. It's much more severe than for a man. I think, Wendy?
Thank you very much. I think you're particularly on target when you are talking about the blurring of the spaces from research to clinical to public health. And that strikes me as raising questions well beyond the genome, as you point out. And one of them that is, I think, interesting to me is the idea of using large data sets, whether they be biospecimens or whether they be data sets. And there has been debate about what, as you say, what normative model to use in the different spaces, and sometimes that may come from turf battles, rather
May come from?
turf battles, or from the idea that well, it's in the clinical sphere, so you should get consent from patients or get their consent to return results. Whereas some want to carve out the public health sphere and say, well, it's public health, and therefore we don't need consent. And so the idea is taking the normative standard as a result of the space you're in. Whereas it may be, because of the blurring, that that's not the issue at all, that you don't need to define the difference between the research and the public health space, for example. What you really need is to identify what particular projects need consent or return, as opposed to what space they sit in. In other words, so my question eventually is, do you think the space defines the normative standard, or is there a normative standard that can be developed independent of the space and may depend upon the question you're asking and the material that you're using?
So it's a great question, and I guess where I'm at, to be really honest right now, is thinking that the space you're in, to use that metaphor, is a good starting point, but that's all it is a good starting point, because then you've really got to dig in. I mean, take, for example, this brouhaha over newborn screening, and then the archiving of blood spots. Some of that just to make sure we're doing it right. We're going to reanalyze and capture. Some of it because maybe we missed a kid and then something awful's happening with the kid. It turns out they do have some metabolic abnormality we should have caught. We've got the blood spot, and so we can go back and look. Some of it to do basic research. So then when you go back and you say, well, what kind of consent, if any? We've had this long conversation, in the public health, legal, ethical community about, is it we just do it? Is it opt out? Is it opt in, et cetera? But really, I think you're right. It's the complex suite of things that we want to do that really should then drive our thinking. And also, I think engagement with people and learning more about what stake do they see in this problem. That should drive our decision about how the architecture of that consent, and by the way, ongoing engagement with people over time. I mean, maybe they want to know what's happening with their kid's blood spot. I mean, I will tell you in Minnesota that for lack of doing it all these babies in onesies that read, the government has my DNA. It was devastating PR. Devastating. So the consequences of failing to zoom in like that and really engage these people can blow a public health program virtually out of the water, as it kind of did in Minnesota. So I think you're absolutely right. We have to zoom in like that. So I'm not sure where we are on time.
We have a couple more minutes.
Couple more minutes? Yes.
Thank you so much. I'm finding myself thinking ahead in this brave new world, and I'm wondering if you ever get concerned more in the clinical space that there will be a time when people are blamed for not having gone through whole genomic sequencing to determine things in advance that they may be susceptible to, such as, well, we could have treated you earlier if we had known this about you. And And is that a question that's arising, and does that at all concern you?
If it were to happen, it would profoundly concern me. I don't see that looming. I mean, on the horizon right now. I mean, one can imagine it.
And I'm really basing it on a lot of things that came before, such as prenatal screening.
Yeah.
It has become very hard to say no to prenatal screening. And will it become hard later to say no to whole genomic screening or sequencing?
Yeah, I know, and I know that literature, and so maybe you're right, but, you know, since we're taping this, why not? I refused amnio. The reason I refused amnio, which may shock you, is because I had gone through a lot of in vitro fertilization to get that far. And I was of advanced age, and I thought, and also the ultrasounds, which now are very sophisticated, didn't indicate that it was needed, but I knew the risk I was taking in doing that, so maybe you're asking the wrong person, you know?
No, I think I'm asking the right person.
This, I guess, is why I cared so deeply about the paper that George and Sherman and I collaborated on, that I think there is a temptation to go, wow, this is the coolest technology, and it's, you know, all this crazy metaphors like the crystal ball, which anybody who does genomics knows that that's just crazy. That's not at all what we've got right now. And it's not really what we're going to get either in this deeply probabilistic, incredibly complex space. But there is this sort of falling in love with, deeply enamored of, that maybe supports the importance of your question. And that's why when ACMG said, you know, you should get people's consent to the basic sequencing, but beyond that, in terms of specific consent to the actual 56 extra or tomorrow 100 extra or 200 extra, no, it's too complicated. It's too complicated for them to understand. I don't buy it. I think that as the technology gets more seductive and more complex, you have to redouble your efforts to explain and create the tools you need. George made the point, and he had made it in an earlier paper, I think, with Sherman. Sure, you can explain it, bin it. Some of this is about cancer, some of this is about cardiovascular, so, you know, et cetera, and I'll zoom in anywhere you want. You can explain it, and you have to explain it, or we will strip people of their ability to make decisions. That doesn't necessarily answer all the public health questions, because look at something like newborn screening, where we've said, well, some states have said opt out, but I don't even remember in Minnesota with my kids anything being said to me. There's supposedly, I think, an opt-out in Minnesota. I don't even remember their heels being pricked. I don't remember a single word about it. So if there's an opt-out, it's a fake opt-out, as far as I can tell, or at least in my, you know, N of one experience. So I think all of this makes ethics very important in this space. Thank you.